Hi, everyone. Welcome to the Learning Salon. We are very excited to be here for our first session of 2023. Happy New Year. We're hoping that this would be a better January 6th than last year. We are very excited today to have Mike Levin. But before talking about that, hi, Jess. Hi, everyone. I just wanted to say a few things for those who are joining us for the first time. The Learning Salon is a online forum for us to discuss contentions and bridges between biological and artificial learning. And we are a very interdisciplinary audience and speakers. As you know, we have people from across AI, neuroscience, psychology, philosophy, linguistics. So if you hear something and you're not sure what it means, you can ask clarification questions in the chat. If you have questions to the speaker or you want to discuss something on the screen or off the screen, you can add to ask a question area below. It's a button, and click on each other, like each other's questions so that the questions with the most likes can be prioritized. Another thing to remember is to not be worried if you're early career, if you're a student, don't worry about asking a question. We have a short format talk and a very long format discussion. So this is a place to get rid of the inhibitions. If you ask something that's off, you can always clarify it either in the chat or come to screen. If you don't want to come to screen, just say ask for me so that we know that we don't invite you. Otherwise, if you're still around, we might invite you. And if you feel comfortable, you can come on the screen and ask your question in person. Otherwise, you can add your clarifications in the chat. A gentle reminder, there's a slight delay. We are a little earlier than what you hear. So some things that you say in the chat, we might see it a little later than you think. So if you don't see an immediate response, it could be because of that. With that, I just want to remind everyone also, disagreements, justified disagreements are completely welcome here because we want to understand. Our goal is not agreement, but understanding exactly of each other's points, whether there is contention or bridges or disagreements or agreements. But disrespect is not tolerated. So if there is disrespect, we will have a warning. And if there is too much, then we might need to have someone exit. So just so that we all know what the rules of the room are, as always. With that, I'm just going to hand it off to John Krakauer. For those of you who don't know me, I'm Aida Momen-Nejad. My name is not here. I'm here as Worldwide Neuro. That's not my official name. And John is going to introduce Mike before Mike starts to share his screen. John, please take it away.
Okay, Mike, I don't think I know we've been on some Zooms together, but I don't claim to know you super well, but obviously I've read your stuff. How does one describe someone like Michael? I've been thinking about this. Well, he's a professor at Tufts, has done sort of seminal work in developmental biology and communication between cells. You can almost see in his seminal early work where he was going in retrospect. I think that he has of late, the stuff that I've been really interested in is really arguing for, cognition all the way down, as one of his articles is called, and also almost something as extreme as saying that life is intelligence. In other words, that you can almost find goal-directedness in any biological system. And I almost feel like that's become his definition of life almost, which I think is really very interesting. Because it makes one feel like intelligence might even be able to uncouple from life one day, which I think is some of the things that he says with respect to all the new synthetic organisms and all the new things that we could be built out of non-biological material and still be intelligent. So I think the field of intelligence by Michael has basically, ruptured and opened to the degree that we may never actually be able to put the genie back in the bottle in terms of how we think about it. Now, I'm not in full agreement with it in its globality, but I certainly think that in a paper that just came out in Nature, I think, saying that science has become less innovative and less interesting and more journeyman, then I certainly think that people like Michael are badly needed, because that's certainly not what he is. I think he's almost a Baroque scientific fantasy and scientist at the same time, and we badly need that. So I think it's, of all the people we've had on the Salon, I think you at least are up there in terms of epitomizing the all the interfaces that we're interested in: AI, psychology, biology, intelligence, life. So you're there in a delicious package, and we're very much looking forward to your talk.
Well, thank you so much for that amazing introduction. I'm so pleased to be here and have the opportunity to share some thoughts with you. I think I will probably say some things even more radical than that. So I look forward to your comments. So let's see. Okay, is everybody seeing my my screen there? All right. So,
Yes.
okay, so here we go. Anyone who wants more details or the primary papers or to get in touch with me, please contact me at these websites. Everything is there, all the data, the videos, everything is there. What I would like to talk about today is this notion of diverse intelligence, and to really kind of abstract from the typical embodiments of intelligence and the places we're used to finding it very broadly, and to really sort of talk about it in a little bit of detail, although time is short. I would like to use the example of morphogenesis, so collective cellular behavior in generating form as a kind of unconventional cognition, and that will be our example. But I really do want to take it much broader than that. So Alan Turing needs no introduction. He was somebody that was very interested in intelligence generically in various embodiments, sort of the essence, right? What is the essence of intelligence? And he was interested, of course, in problem solving machines and intelligence through reprogrammability or plasticity. You may know that he also did something quite unusual. He wrote a paper on morphogenesis. And you might wonder why somebody that's interested in intelligence and computation, why would somebody be like that be looking at morphogenesis? And I think that he saw a profound symmetry between these two processes, which is only now beginning to be appreciated. And I think Turing appreciated it, although he didn't write directly about it. One interesting thing is that people often draw a distinction between things we call collective intelligences, so ant colonies, bird flocks, and so on. And so we can debate whether these are collective intelligences or not. But then people say, well, those are sort of weird collective intelligence, but I am a true integrated, unified intelligence, right? I'm not a collective intelligence. And I want to say right out of the gate that actually all intelligence is collective intelligence. So there is no, to my knowledge, there's no such thing as some sort of indivisible diamond of intelligence, some sort of monad that has no parts. We are all made of parts. And so we all need to understand how our intelligence emerges from the interaction of these parts. Now, what do our parts look like? Well, here's one example. This thing is called a lacrimaria. So this is a single cell. It has no brain. It has no nervous system. It has no cell-to-cell communication within its body. It's one cell. So you can see what it's doing here. It's managing all of its physiological, anatomical, behavioral, metabolic needs at the level of a single cell very nicely. It has incredible capabilities of body control. Soft body roboticists sort of salivate at the sight of things like this. We have nothing like it. And because we are all made of single cell creatures, meaning that all of our parts used to be individuals themselves before they joined up, one important thing we need to start to do is to dissolve this. So this is a kind of pre-scientific view that is really focused on natural biological kinds. There are species, there are humans. We all know what a human is. We all know what animals is. They come in various types. This whole picture has to go because of the plasticity and interoperability of life. And I'll get to this towards the end of the talk. So the standard view is that there's something like this. This is a standard human. It's got a sort of glow, this like agential glow about it, where it's okay to talk about this kind of being as having memories, goals, preferences, and things, and so that's okay. But that glow doesn't really penetrate very far because when you start to talk about other kinds of creatures, one is sort of accused of anthropomorphism and things like that, as if this is a distinct natural category that has these properties that nothing else has. And so we want to start thinking about what does it mean to be a self as the owner of memories, of preferences, of goals. and of competencies to solve problems in specific spaces. So let's think about this. So here's a brain. If someone showed you this particular anatomical structure, 3.5 whatever pounds, if you didn't already know what it was and what it's like to interact with the inhabitant of this thing, how many selves would you say should be in there? We sort of take for granted that, well, there's typically one integrated self in there, and we assume, well, of course, it's the brain, so there's one person. But actually, it's completely not obvious how many should be in there. How many cells should there be in a set of neurons this big? And interestingly, I will claim that is exactly the same question as asking how many embryos are going to appear out of a blastoderm of cells. So we all start life as this kind of sheet of cells. So let's say there's 50,000 cells. And within that 50,000 cells, we are going to assemble one embryo. Normally, that's what happens. You assemble one embryo. And so a couple of interesting things. In both of these cases, you cannot guess in advance because it is not determined actually by the genetics or by anything else like that. It's determined by a physiological process of self-organization that will give rise to one major self or individual in these cases. And then you might wonder, well, what are we counting? When we say that there's one cognitive individual in the brain, there's one that comes out of the blastoderm, what is it that we're counting? Well, what we're counting is the ability of some section of this like Freudian ocean of possibility to self-organize into an agent, and then we have to define this. And I'm going to give you my definition of what a self or an agent is. But it's really important to know that in embryogenesis, what we could do, and we've done this, as have many other people, you can make temporary scratches in this blastoderm that prevents these regions of cells from talking to each other. Then each one of them organizes into a separate embryo, and that's how you get conjoined twins. And in that case, there will be some disputed zone, but there will be regions of embryos. And again, one starts to wonder, what is it that we're counting when we're counting these embryos? Because that blastoderm can give rise to 0, 1, 2, 3. We don't know exactly how many, but that number is not determined at the time of the egg fertilization. Now, the same thing is true, of course, in cognition, right? So we understand split brain patients. There are dissociative disorders. The question of how many selves are in these dynamical systems is open. And in order to try to understand this, where do these selves come from? We have to think about this amazing journey that all of us took from what people call just physics. So here's the unfertilized quiescent oocyte. It's a little pile of chemicals. They're not doing much. Everybody looks at it and says, okay, there's no agency there. There's no goal-directedness. This is just physics and chemistry. There's nothing else. And then we slowly but surely undergo this incredible magical process of embryonic development where we end up as one of these things, or perhaps as one of these things that will then something like an adult human, perhaps Rene Descartes will say that, okay, well, I have something that this thing does not have. Now, the key is that developmental biology studies this transformation, and every part of this is slow and gradual. There is no place, developmental biology offers no place for a magic lightning bolt that turns this into this. So whatever it is that happens here, and we can talk about phase transitions and things like that, but whatever it is that happens very slowly and gradually. And so this continuity thesis asks us to think about, where does this Cartesian cut come from, right? Because we can turn the clock back minute by minute, literally from this all the way through this. And if you're sort of tempted to say, well, at this point there is no cognition, and then at this point there is, the next question is, fine, show me what happened, right? What led to this? What change led to this? There is nothing in development that offers a convenient point. So what I'm interested in is counting selves, which are goal-directed, integrated, self-organizing systems with various degrees of competency in arbitrary spaces. So a self, you need to do 3 things to identify a self. You need to find some problem space within which you think that self lives. You need to make some sort of claim about a boundary between that system and its outside world. And you need to make some kind of a claim about the cognitive light cone. Specifically, we'll get more to this, but specifically the size and type of goals that system is capable of pursuing. So I want to talk a little bit about some biological examples of that plasticity. You know, this all sounds needlessly baroque. I mean, we kind of all know what individuals are, right? The barrier, you know, at the edge, we all stop at the edge of our skin, we think, and so on. But I want to really focus on the plasticity. So here are a few examples. So here is a caterpillar. This animal lives, it's a soft-bodied kind of creature, so soft-body robotics are relevant here. It lives in a two-dimensional world. It crawls around, it chews plants. What it needs to do is transform itself into this animal, which lives in a three-dimensional world, doesn't care about the leaves at all. What it wants is nectar, and it has a completely different brain. During this metamorphosis process, most of the cells in the brain are dissociated from each other. Most of them die, in fact. The brain is reassembled fresh in a completely new architecture. And yet it's been shown that memories that form in the caterpillar, when you train the caterpillar or larva with some memory, the moth or butterfly still remembers. So we can think about, what does it mean? Well, two things. One is that what does it mean for information and knowledge to survive radical rearrangement of its substrate? That's first. And second, you know, if in Philosophy 101 we think about what's it like to be a butterfly, this is that question on steroids. What is it like to be a caterpillar turning into a butterfly? So we have to just realize that our cognitive systems are strongly embodied, and our bodies are plastic. So here's another example. These are planaria. I'll show you more of this later. These guys are amazing because they regenerate every part of their body, including their brain, which is in their heads. So what you can do is you can train these planaria on some particular task. You can cut off their heads. The tail will sit there doing nothing for about 8 days. They then slowly rebuild a brand new head. And then then whatever information existed in the rest of the body is imprinted onto this new brain, and the animal remembers. Now, of course, you can cut it into many pieces. The record is something like 275 pieces. And all of these guys will rebuild perfect, tiny little worms. So this old philosophical experiment about what would happen if you were transported, that's fine, but the transporter malfunctions. It makes a copy of you, but leaves the original intact. Which one are you? Well, here is that experiment. Which one of these planaria are the original? Right? So there are many cases in biology like this that really start to dissolve this old simple idea of you know, we know what the brain is. It's kind of a fixed structure. We study its capabilities, and we work on this notion that in biology, not only are we nested dolls of structure. I mean, that much is obvious, but each level has its own competency. So we call it a multi-scale competency architecture. The molecular networks, the subcellular components, organs, tissues, and then of course swarms, all have varieties of problem-solving abilities in different spaces. That's what we're going to talk about today. And what I've been working on is a framework called TAME, T-A-M-E, stands for Technological Approach to Mind Everywhere, which really seeks to help us be able to recognize, create, and then relate to truly diverse intelligences. So not just familiar creatures like us, apes, birds, maybe octopus, but really weird things like colonial organisms, swarms, new synthetic engineered life forms, engineered into AIs, you know, software agents, and then potentially exobiological species. And so I follow this sort of idea from Rosenbluth, Wiener, and Bigelow, where they laid out this very cybernetic kind of way of looking at cognition that really shows you that it comes in degrees. It's not a binary. You know, I really don't like binary categories. Yes, this is cognitive. No, this is not. I think we're looking at something like this, where you can really start to flesh out categories of capabilities from very passive kinds of systems all the way up through very complex metacognitive human systems. And so the goal of this framework, of course, is not just philosophy. It's to enable new experiments. So what we've been doing with this idea of continuity thesis is to say, all right, what if could we come up with a definition of intelligence and cognitive competency that is substrate invariant, that does not have to be the typical embodiments that we're used to, but actually could be looked for using all the tools and techniques of behavioral science and computer science and so on, in weird, unconventional substrates. So the thing is that so here's what we're very good at as a species. We're very good at recognizing agency of middle-sized objects moving at medium speeds in the three-dimensional world. So, when these crows do various things to solve problems, we all recognize that as intelligence. But that's because all of our sense organs are pointed that way and have been focused that way. Imagine if you had a built-in ability to sense your blood chemistry, the way that you are able to see and feel. Imagine if you really could sense your blood chemistry. I think we would have a very good intuitive understanding of the intelligence of our liver and kidneys and other organs, because you would be able to see the moves that system makes in physiological space, given various novel things that happen. So there are many spaces we're interested in: the space of all gene expression, the space of anatomical patterns, the space of physiological states. There are many systems that navigate these spaces with the kinds of competencies that psychology and behavioral neuroscience study. And so we want to understand it. So let's look at my favorite, which is the anatomical morphospace. So we all start life as this collection of cells, these blastomeres, and this is a cross-section through an adult human torso. So look at the amazing order of all these organs. Everything is in the right place, the right shape, orientation with each other, and so on. And so where does this anatomical structure, where does the information reside? Now, you might be tempted to say DNA, but we can read genomes now. And when you read a genome, you don't see any of this. What you see is a specification of the micro level hardware of every cell. In proteins, you see protein sequences. And so the big challenge of developmental biology is to ask, given that hardware, how do these very active little components know what to make, when to stop? If it's missing, workers in regenerative medicine, like my group, has to ask, well, what do we say to these cells to get them to recreate it, repair it? And as engineers, we ask, well, what else is possible? Never mind this default state. What else can we get the exact same cells to do? So we're studying this, the collective intelligence of these cells using roughly the definition of William James, which is the ability to reach the same goal by different means. As someone else put it, and I wish I could remember who said this, because I think it's a great saying, the spectrum between two magnets trying to get together and Romeo and Juliet trying to get together. What's the spectrum? The spectrum is the degree of diverse competencies, starting with the ability to actually step away from your goal in order to sort of get close to it later on, which magnets won't do. The spectrum of competencies you can expect from a given system in fulfilling its goals. So having focused it that way, you can ask about the cognitive light cone. If we collapse time and space into one axis, you can ask, how big are the goals of any given system? Is it local sugar concentration? Well, you might be a bacterium. Is it the size and shape of an organ? Well, then you might be a liver, and so on. And then, of course, if you're a human, you could have massive goals that are planetary scale and in terms of time go beyond your lifespan. So what kind of intelligence do cells have? Now, one amazing thing is that they're able to make the same anatomy from different starting states. So you could take an embryo, cut it in half, and you don't get two half bodies. You get two perfectly normal monozygotic twins. It's able to adjust for that. And in general, what they do is what living things do is they move through morphospace, the space of possible anatomical configurations, with the ability to start off in different positions, have various barriers, local optima like this, and still get to where they're going. That, of course, is the basis of regeneration. So here's the salamander. These axolotls regenerate many of the organs of their bodies. I can amputate this limb anywhere, and the cells will build rapidly. They divide, they undergo morphogenesis, and then they stop. When do they stop? They stop when a correct salamander limb has been completed. That is when they stop. So this is an example of a kind of anatomical homeostasis. There's a set point error, that's deviation from the set point, triggers a massive activity of this collective. Individual cells have no idea what a finger is or how big the arm is supposed to be, but the collective absolutely does, because as long as you keep deviating it, it will keep going and do some very interesting things to get there. Here's an example of creative problem solving. This is a tadpole of the frog here, the eyes, the brain, the nostrils. This thing, in order to become a frog, has to rearrange its face. And so in order to do that, all of these different things have to move around. And so what we did was we created these Picasso tadpoles. And so everything is in the wrong place. The eyes on the back of the head, the jaws are off to the side. And we found that these guys still make a pretty normal frog face, because everything doesn't just move in a hardwired pattern. Everything moves in a novel path as needed and keeps moving until it makes a correct frog face, and then it stops. So it's got the ability to make up for very novel circumstances. So this is, long story short, I don't have time to really go into it, but we've been studying the isomorphism between how goal-directed behavior happens in the brain, in which there is an electrical network that binds individual neurons to common purpose. The exact same thing happens everywhere in your body. It's a much more ancient system. That is where the brain sort of learned all of its tricks. And so electrical networks bind the cells in your bodies together into systems that can store pattern memories as anatomical set points, representations of what it is that they're going to build. How do we know? Because we've developed tools to rewrite those memories. So much like people in neuroscience try to incept false behavioral memories into, let's say, mice, we can do the same thing in the rest of the body. And when you do that, you can do things like this. We can, so here's our frog, our tadpole. You will notice that it has an eye on its gut. That's because we took some cells and we introduced a particular RNA that creates an electrical pattern that says, you should be an eye. Not only do they make a perfect eye with all the same lens, right, and all that stuff, but in fact, if you don't hit enough cells, the blue ones are the ones that we've hit, they actually recruit their buddies, very much like ants will recruit others to some task. They recruit their unmodified, completely wild-type neighbors to participate in making this nice lens out on the tail somewhere. So they've got this competency of recruitment. We can take this perfectly normal one-headed worm and rewrite the electrical pattern. We don't do this by applying electricity. We do it by targeting ion channels. And instead of a one-headed pattern, we can say make a two-headed pattern. This is a map of this anatomically normal animal. So nothing happens. This memory stays latent until it's injured. Once we cut the two pieces off, it will then rebuild whatever the pattern says. This is so we now have direct read and write ability into the cognitive medium that underlies the collective intelligence of these cells as they navigate anatomical space to get from a cut fragment to a correct worm. And by the way, it's a counterfactual memory because it's a memory of something that isn't true now. This animal thinks that planaria should have two heads, but it's got a one-headed body. This is not the case now. It's not a readout of a two-headed animal. It's a future state that it's storing. And if you do this, like any good memory, it persists. So these two-headed animals stay two-headed as you keep cutting them with no further manipulations. Remember, their genomes are wild type. There's nothing wrong with their hardware. This is a in software change. And here you can see the video of here are the two-headed worms. So this, I sort of floated this idea of the size of your goals. So this has a very actionable, and this is I'm almost done. I just have a couple of slides. It gives you a very actionable path towards some really important biomedical goals, such as cancer. So we know what evolution has done. Evolution has enlarged the cognitive light cone of systems to go from tiny little single cell goals, metabolism, reproduction, to very large goals like make a limb. Yeah, the system is able to pursue a much larger goal. But that process has failure modes, and sometimes it breaks down and cells defect, they electrically disconnect. And so this is human glioblastoma. They basically just treat the rest of the body as external environment. They're no longer attached to that computational network that knows what a limb is and how many fingers it's supposed to have and so on. So the last thing I want to show you are some artificial organisms. So we asked the following question. We wanted to know if we make a new creature that's never existed on Earth before. We're using biological cells, but we're putting them in the new context that has never been selected as such. We want to know how much creative problem solving exists in that context. And if there is any, what are the goals that these anatomical, behavioral, and so on, that they will have, given that they were not shaped by evolution? And I have to do a disclosure here. Josh Bongard and I are founders of a company called Fauna Systems, which works in computer designed biorobotics. So here's what we did, and this is the work of Douglas Blackiston. He did all the biology, and Sam Kriegman did the computational work. What we do is we take the skin cells off of a frog embryo and we put them aside, and they could do many things. So here they are. They could just die. They could spread out and then go away from each other. They could form a two-dimensional monolayer. Instead, what they do is they coalesce into this amazing structure we call a xenobot. So xenobots use the little hairs on their skin that are normally used to redistribute mucus. They use the hairs to swim along. So you can see this guy chugging along here. They can go in circles. They can patrol back and forth. They have collective behaviors that you can see here, them interacting in various ways. Here's one doing a maze. So this is kind of interesting. This is still water. There's no flow, no gradients. It goes this way. It takes the turn without having to bump into the opposite wall. At this point, for some internal reason we have no understanding of yet, it decides to turn around, go back where it came from. If you scan them for calcium activity, it's very brain-like. This is what you see when you look at calcium activity in brains. There are no neurons here. This is just skin. This is literally, his whole organism is made just of skin cells. And they have an amazing capacity, because what we've done is made it impossible for these guys to reproduce the way that normal frogs reproduce. Well, if you put them in an environment with lots of loose skin cells, what they will do is basically von Neumann's dream. They become a machine that assembles copies of itself from parts it finds lying around. So what the bots do is they run around and they collect other skin cells into these little piles. The piles, because they're working with an agential material and not passive particles, overnight the parts themselves, these little piles themselves, become the next generation of Xenobots. And guess what they do? They run around doing the same thing. And so you have generation 0, 1, 2, 3, and so on. So they have this, we call this kinematic self-replication. So here's the amazing thing about this. If you sequence these Xenobots, all you see is standard Xenopus laevis genome. There are no genetic changes here. And under standard circumstances, what these cells will do is produce this kind of embryo with this kind of tadpole behavior. And you might think that having this really boring two-dimensional life on the outside of the animal, sitting there passively keeping out the bacteria, is the only thing that these guys can do. But liberated from the behavior shaping signals of the other cells, we don't give them new genes, we don't give them nanomaterials, nothing. All we do is we engineer by subtraction. We take away the things that are normally basically bullying these cells into just having this like quiet, boring life. They become a Xenobot. They have their own developmental sequence. It looks like this weeks into. We have no idea what this is. None of this ever existed before on Earth, as far as we know. They have new behaviors, such as kinematic self-replication. And what you can't do with systems like this is the typical go-to move when you ask someone, why does a certain animal have certain behaviors, certain anatomy? Well, they say, well, years of, eons of selection, right? It was always selected to be this or that. Everything else died off. Well, there's never been selection to be a good Xenobot. There's never been any Xenobots. Why is it that within 48 hours of finding itself in this new circumstance, you get a panoply of developmental stages and capabilities? By the way, we don't know what their cognitive capacities are. We're of course finding that out now. Can they learn? What are their preferences? We don't know. So the final thing I want to leave you with is this idea that when Darwin said endless forms most beautiful, he was really impressed with the diversity of life on Earth. All of that is this tiny little corner here of the space of possible beings. Every other combination of evolved material, engineered material, and software, so hybrids, cyborgs, every kind of combination, because life is so interoperable, and we can talk about why that is. All of these things are viable. All of these things are going to be coherent. They're going to have various competencies in various spaces, not just the familiar three-dimensional, space of classic behavior. And all of this has massive implications, not only for intelligence and plasticity, but ethics and ways in which we're going to relate to systems that do not share with us any position on the phylogenetic tree. And perhaps nothing is, no commonality in terms of where did you come from and what do you look like, right? And we have to really develop frameworks for relating to those creatures. So this is it at the end. I just want to show you this is a great video of a very young cat figuring out that it's got paws for the first time, and it's very surprising, of course, because as an agent, when you are assembled from cells into the world for the first time, you really don't know where the boundaries are. Every cell is some other cell's neighbor. You have to build a model of yourself, of the outside world, of some sort of boundary between it and the outside world. You are under severe energy constraint, so you cannot track microstates. You have to coarse grain and have symbols for yourself and for other important things in the environment. And you have to do all this from scratch, basically, because you don't know how many cells you will have, who's your neighbor. You don't know any of that. So this is it. This is I think what's really important is to understand the origin of competency in diverse spaces, and that has implications for AGI and things like that. So that's it. I would just like to thank the postdocs and the students who did the work that I showed you. Again, 2 disclosures: Morphoceuticals is a regeneration company that I'm associated with, and then Fauna Systems. And I thank you for listening.
Thank you so much. A lot of questions and clarifications. I'm not sure if I should start or if maybe someone else should start. Maybe I'll ask like a couple of clarification questions. I don't know if it's just me, but it seemed to me like there were some analogies that were too far, and the steps in between weren't scientifically justified for me. A number of those analogies, I thought, sorry, there's a siren, classic New York City. Yeah, are you suggesting, for instance, in the last example, that first cats have selves, and are you anthropomorphizing the intentions or perception of the cat for showing that behavior? It's just, even in that example, it was a little too far. Another case where it wasn't clear to me and it seemed it was a little far was when you said the frog skin is brain-like. Did you just mean a loose metaphor about visually having signals, or that you have some measures of minimal computational features that would make a tissue brain-like? I didn't see the latter, so I'm not, I feel like all of it is extremely interesting. I'm super excited about the ideas, and I talk about it a lot, of expanding the notion of intelligence to be not just human-like intelligence, but I think you're doing something else. You're saying all of it is human-like, and I think I disagree with that. I think that the metaphors for me just don't hold water scientifically, so with evidence, so to speak. So maybe we start with that clarification, then I'll hold my questions until after John and Melanie.
Sure. Yeah. Of course. So let's just start with the fact that I tried to keep this to 20 minutes. So there's absolutely no way that I can stick all of the scientific background into all of this, right? So keep in mind, we run a lab focused on regenerative medicine. So none of these analogies are of any use to us unless they're producing new capabilities. So the criterion for everything I say is that it gives rise to new capabilities at the bench, and we've had many of these. And so the point is not to produce philosophy. Everything I've said, with the exception of the cat at the end, that was more for fun. I have no claims about cats whatsoever. Okay. But everything else has very specific. The framework that gives rise to this has given rise to treatments for birth defects, ways to address cancer, regenerative. All of that is because these things have specific scientific implications.
Sorry. So let's just not. So one thing is the scientific or engineer. It seems like it's more engineering implications of something that you're doing. Another thing is, and we're not talking about philosophy. Scientifically, how do we say something is brain-like? There needs to be some computational specific tests to make that claim. Sure. And there needs to be some kind of work to be done to make that claim. Was that also like the cat just a loose metaphor? Because whether it produces like drugs or not that are helpful or some other thing, it doesn't speak to the scientific claims. I'm asking about the scientific claims, it's not the outcome of you thinking like this or calling it that, because I think even if you didn't call it that, the outcomes would happen. You don't need to.
So let's talk about that. I think that's absolutely false. So let's talk about that. So two-headed planaria. Two-headed planaria were seen for the first time in 1904, roughly. For 100, so not until 2009 did anybody ask what happens if you recut a two-headed planarian, because it was completely obvious that your shape is an emergent outcome of your genetics, and that given a wild-type genetics, all of those cells should only be able to remake two-headed planaria. By asking the question, could techniques from neuroscience, meaning things like optogenetics, things like targeting memory pathways, the things that are used on minimally cognitive agents having memories and implementing those memories in some space, could that be relevant to development? That was never thought. So the idea that actually by porting real tools. So everything from active inference, serotonergic signaling, all of these things that are used in very specific ways in brain science. What we've done was to use them in a completely different context by considering the hypothesis that what development is a walk-in morphospace by a set of cells that use very brain-like mechanisms. So we're talking the same genes, the same algorithms, the same information processing. Like literally, we steal everything from neuroscience and apply it to things that are not brains at all, to be able to reach novel phenotypes and novel, better predictive frameworks that never existed before. So there's a reason why everything from neuroscience works perfectly in these other contexts. That the techniques and the concepts cannot tell the difference. It's exactly the same, and you don't get there if you don't believe that these kinds of concepts. So of course, I never said that any of this was human-like. The point is not that it's human-like. The point is we are not magic. Humans are not magic. We have some cognitive capacities, and those cognitive capacities have an evolutionary history. And we can take advantage of these techniques by studying the more primitive versions.
Just to be clear, using methods from neuroscience, applying it to a different domain and achieving success doesn't make the new domain brain-like. It just means methods developed in neuroscience help something. For instance, at Microsoft Research, we might use methods from neuroscience in order to analyze something else that has nothing to do with brains. That is a way of exporting techniques across disciplines. But
if enough of those methods work, then I'm not sure what you mean by saying it's not brain-like. So if I can use serotonin,
then let me mention that scientifically, what I'm saying is that for something to be brain-like, it's not enough to say I had engineering success by deriving methods from the other one. Why? Because the mathematical, computational, data analytic reasons why those methods worked are not clear. What is the actual conceptual scientific reasons why those methods were successful in another domain? It could be exactly that there clear. Sorry, let me finish. Here we don't kind of, no. For instance, let's say that you applied something like, I don't know, let's say you applied something from neuroscience, a particular approach from neuroscience in manifolds that we capture from the hippocampus to analyze behavior somewhere else. Does that mean that that kind of behavior that you observed was generated by a hippocampus or something like the hippocampus? No, it just means some method that we applied that worked for the hippocampus can work for this other data too. And the reason that might work could be some mathematical principle, some computational principle, like the particular manifolds, the particular multi-scale hierarchy that there is in both systems. It doesn't mean that one of the systems is necessarily in the same category as the other. It doesn't make anything that analysis developed in the hippocampus can apply to. Anything that can use that successful in engineering way is not a hippocampus, just like that. Likewise, if something is applied that worked in neuroscience at different levels of neuroscience to something that was much more closer to the cells working somewhere else, it still doesn't say this is brain-like. It says something that we applied in neuroscience worked in this other domain. I think the principles for actually claiming something is brain-like should be much more clarified, much more clear. What is the minimal criteria of a brain? It should be shown across different brains. It should be established before one can make the claims. I think maybe we disagree on the level of precision needed to make these claims.
So let's put it this way. Of course, I'm not saying that any of this is like a hippocampus, but I personally have no interest in the hippocampus at that level. What I'm interested in is conserved general dynamics that make the brains interesting. And those are particular kinds of information processing and capabilities that we find in other systems. So we're certainly going to find various degrees of intelligence and competency in let's say exobiological agents, different kinds of chimeras and synthetic agents. And to me, it's really not very interesting at all to try to say that this has exactly the same brain architecture. What I want to know is, how does it solve problems? How does it process information? And so, and then the judge of all this, right? So how do we discover whether some particular some particular invariant is worthwhile or interesting, is what level of prediction and control it affords you. And so my point is simple. By importing concepts, not specific structures of hippocampus and things like that, but by importing specific theories of how collectives of electrically active cells process information about past, future, integrating across space and all of that. The fact that the brain didn't invent these tricks from scratch, of course, there's an evolutionary history to this, and so in fact it goes all the way to bacterial biofilms. Even bacterial biofilms do some of the same tricks that we see in nervous systems. So that to me is the interesting part, right? Not the specifics and the sort of comparative neurobiology of brains across species, but what is it that's interesting about these? And why do we care about brains, right? What are they doing that's interesting?
So so just to recap what I understand what you're saying actually your claim is not that something is brain-like. Your claim is that there might be some computational principles that are in common across various kinds of cells. That is a claim I'm totally on board with. I'm also very much on board with the idea that there are some collective dynamic principles that are in common between collective entities like swarms versus, let's say, cells in a brain or cells in a body, and therefore there are some comparisons that might be possible. But I think where I disagree with you is the extent to which the claims go. And then I would say perhaps the imprecision with which some of the analogies or claims are drawn here. It kind of makes me uncomfortable. It's like, no, we haven't shown this. We've shown maybe that, or not we, you, maybe you've shown that some of these approaches in neuroscience can sort of help in this other domain. But the specific claim that this is brain-like, that's not the case. Or the cells versus embryos, the specific claims that there is a similarity between cells and embryos, or cells and cats, like all of those analogies where I don't know where you're making a claim, where you're saying later it was a joke. It's kind of a little uncomfortable for me, but I'm going to wait for John and Melanie to ask their questions. And then if these things get clarified, maybe we can continue this. I just find that the level of claims, there's very interesting work, amazing work, and I'm very interested in it. But it's very distracting for me to see the way that claims work. It's just all the claims don't seem justified to me. That's the issue I'm facing right now. But I'm going to stop so that John and Melanie have some time. John, do you want to go first?
Sure. Just to follow up for a second on what Ida's saying. I do think it's actually true, Michael, that you do. And it's fun, and I can see why we should all do this. It's like, everyone who's creative, is it you do tend to let extrapolation slip in that I think do go a little further, to my mind, than all the examples in their Baroque, fantastic nature justify, right? And first of all, let's start from the beginning. If I were to show that very similar differential equations work for the decay of an isotope and for predator-prey relations, and I can write out very similar differential equations and just put different contents into the terms, it'd be very weird to say, therefore, predator-prey relations and radioactive decay share some fundamental mechanism because you can describe them with the same differential equations. That would be a very odd statement, and that seems to be what you are saying, that because you can see some compressed similarity for seemingly disparate things, therefore they're the same thing in some way. And I just don't think that necessarily follows, right? I mean, it would be very odd unless you do believe that the decay of an isotope and predator-prey relations are at the level of mechanistic substrate similar.
So yeah, so a couple of things there. What do we mean by similar? What I mean by similar is that that we are trying to identify a fundamental way of thinking about these systems that will help us in prediction and control. So if your equations work correctly in this other system, mechanistically they may share nothing. That's fine. but they must share something, and that something may be more interesting or less interesting, right? In terms of if they literally shared nothing, your equations wouldn't work. They must share something. And so now the question is that something interesting, useful, whatever? And so let's just this idea of forget the brain for a second. Let's talk about neurons. So, and let's just take one single neuron. It's in certain contexts. If I were speaking to a group of neuroanatomists, of course I wouldn't make the claim that we don't know what a neuron is. Of course we know what it is. We know the different types. Here they are. Fine. But more broadly, I've been in evolutionary biology meetings where we spent 3 hours arguing about what a neuron really is. Why? Because if you work it backwards, you find cells all the way back to bacteria that once you've sort of written out the criteria of what a neuron is, you find the same things. And in fact, Koshland wrote some of the first neuroscience papers by studying electrophysiology in bacteria. You find some of the exact same dynamics. So is a bacterium a neuron? Well, for the purpose of a neuroanatomy class, absolutely not. For the purpose of asking what's interesting about a neuron, actually, and what's interesting is a particular set of electrical dynamics that allows it to process information in networks with other such things. That is one thing that some people find interesting. You may not, and that's okay. And so I think a lot of this is dependent on what your goal is and what the context is.
Well, exactly right. But if you open your reframing cognition, getting down to biological basics, opening to the proceedings papers, and it gets to precisely this, right? You say that you need 3 essential steps. The cognitive sciences should at last join the rest of the life sciences in the way they approach their quarry. This involves 3 essential steps. First, start with the smallest and simplest organism that display the phenomena of interest. But displaying the phenomena of an interest is the entire thing. How do we agree that it's displaying the phenomenon of interest? Now, that presupposes that you know what that similarity criteria is. So the argument's kind of circular, right? Because you've already decided what the thing is that you're going to claim similarity for. So in the case of the differential equations for prey-predator relations and decay of isotopes, you can decide that you think, that they both are subject to similar differential equations is really the similarity of interest. Well, then you're going to have to tell me beforehand, a priori, why you think that similarity is of interest, and what toolkit of concepts are you going to use a priori to say that that's the similarity of interest. And you're going to have concepts that you've brought from somewhere to justify that similarity. So unfortunately, there's something, it's not quite circular. It's kind of a little bit of a cheat is what's going on, because you're presupposing that you'd have the criteria before you start for the similarity of interest. And then we're right back to square one.
So what I would say is this, and Chris Fields and I have spelled this out in a couple of papers. I don't believe that there is an objective single perspective on any of this. So what I argue for is an observer dependent lens. So if you want to say that this particular system has a level of cognitive capacity, you, exactly as you just said, I agree with you 100%. You have to make some claims. You have to say, what's the problem space? What's the agent? You have to demarcate the agent. You have to make claims about its capability. And then you have to do the actual experiment. None of this is a priori. You have to do the actual experiment and show whether or not all of the things that you've claimed have gotten you to a better understanding of the system. Someone else may look at it with an entirely different lens, pick a different, they may say, that's not I don't think that's the goal it's following at all. I think it's doing this or that. I think it has more or less capacities than you said it did. And then we can compare, we can say, okay, here are 2 worldviews. Here are 2 ways to look at the system. Which of these things, and maybe both equally, which of these things are useful to us? And so all of this starts with a set of claims. And then the question is very simple. What did those claims allow you to do? And all I'm saying is, there is a particular lens on these phenomena, and there are other lenses. I'm not claiming this is the only way to look at these things, but I'm saying that if you look at them in this way, you get to see things and do things that were never done before. That's it.
So let's get to that then. Okay, exactly. So I don't have a problem as I just said, with seeing really wonderful self-organizing behaviors, goal-oriented. I mean, I've always, as a doctor, I've always been amazed at, you lose half the liver and then it regenerates directly back into the right space. And it is amazing. And you have some beautiful things in your paper, the main entropy paper, where you talk about this sort of organizational principle that seems to only express itself at the level of the collective. I mean, it's extremely interesting that those kinds of examples. And, I can give examples in neurology, right? You get this movement disorder, torticollis, where you involuntarily, contract your sternocleidomastoid and to turn your neck, and then people inject into the muscle to stop it, and other accessory muscles take over the goal of moving your neck. So in other words, you've got this allostatic system that has a goal, and it will fight you're trying to, and Peter Sterling's written beautifully about this in his arguments about allostasis and what is health. I'm sure you're familiar. So, abnormal reflexes, you have a spinal cord injury and you end up with all these unusual, highly organized behaviors, spasticity, reflexes, synergies that were never there latent until you had the spinal cord injury. And so, yes, you've got these unwanted, latent, organized behaviors that reveal themselves when you've taken away some top-down control. So there are endless examples of structured, organized, seemingly goal-oriented behavior in subsystems. And that's all great, right? But it seems to me that, and my brother David's talked about this, when is something a model for something and when is it a metaphor? And what I worry about is that a lot of your behaviors that are seemingly goal-oriented, like a liver's goal is to regenerate to a certain size, the word goal, to Ida's point, begins to sound like a metaphor. My goal to go to college and the goal of a liver to generate a certain size, that is a very stretched abuse of the word goal. And the thing is if you were just interested in looking at through the goal-oriented lens at all the different behaviors, because it helped and did work, like you say, that would be great. But let's be very clear about your ambition. You say in the article you made us read, bacteria swimming up nutrient gradients, moths' repeated attempts to reach a light, and human goal-directed behavior must be on the same continuum, because modern biology offers no discrete magical event that separates them. A single framework is needed. Now, quite frankly, that just seems extremely odd. If I were to say, walking, scurrying, slithering, and flying are all forms of animal translocation. And translocation is going from A to B. So those are all translocation. So I'm going to come up with a single theory of translocation, which will basically turn flying and walking and wheels on a car all into the same thing. I just don't buy it. Flying and walking and slithering and wheels are very different things. And saying they're all translocation doesn't really get us anywhere unless you want to do physics. So it seems to me that you've got something so abstract and so all-encompassing, a la Friston, that differences just go away. And by the way, when Friston was asked exactly this question by Sean Carroll, okay, so a bacterium, a cat, and a human, what are the differences that make a difference if they all follow this general translocation rule? And you went, when does a single grain of sand become a pile of sand? He basically said, I have no idea. In other words, the conception is so abstract that the differences that we actually care about, which is, I think, what Ida's getting at, there's nothing to say about them. Right. So in other words, what I really want to know is if you want a theory of everything, which you explicitly say you do, I'd like to know how all these examples of metaphorically using the word goal and showing at some level of abstraction that they're translocating is really going to explain the difference between a human wanting to go to college and a liver wanting to generate to a certain size. I just don't see that theory of everything.
Yeah. So I would say two things. First, I want to talk about a very specific example of where exactly that helps, and then I would say that in the absence of such a theory, then what the alternative view owes is a story about how one becomes the other. If these are not examples of the same thing, then where do human goals come from? And of course, many people have claimed to have such a theory. But when you sort of try to track it back to embryonic and evolutionary past, again, it's very unclear. I've never seen a good theory for where these things pop up. But let's talk about a very specific example, right? Why does this unification matter? The thing about goals, the fundamental thing about goals, let's take the simplest possible goal directed kind of system, a simple homeostat, like a thermostat. Okay. The one thing that's common to all such goal directed systems is that they have an explicit set point that is encoded somewhere. What that enables is a particular kind of relationship to that system, an intervention. It means that when you want to modify that system to change what it's going to do, you don't have to rewire the hardware. You can leave most of it intact, and all you have to do is you have to edit the set point. It has an explicit set point. That's a very particular kind of manipulation that you cannot do with most physical systems. You can't do it with mechanical clocks. You can't do it with many systems. You can't do it, but with your thermostat you can, and with many other systems you can. You change the set point. So here are two competing worldviews. One worldview says morphogenesis is a feed-forward emergent process. There are no goals. All that happens is that there are individual cells operating according to local rules. There is some kind of complexity, cellular automata thing going on, and eventually, whatever happens. They end up, in this complex pattern, right? And we know many kinds of simple rules that give complex patterns. Okay, that's one worldview. Another worldview says, what if? Here's the hypothesis. What if the system is actually a homeostatic system? What if it has an actual representation of the goal state towards which it is trying to develop? And what if it literally has that error minimization scheme where it compares current state to the set point and tries to make that error as small as possible? That second worldview makes a very specific prediction. The prediction is, well, then, you ought to be able to rewrite that set point. That is never going to work with a system that doesn't have an explicitly encoded goal state. So now so now we do an empirical experiment. The first model says there is no encoded goal state. The second one says you should be able to find it, you should be able to decode it, and you should be able to rewrite it. And then, without any change of the hardware, without any genetic change, you should be able to get the cells to build something completely different. This is exactly what we've done. So in the case of planaria, we actually found a standing bioelectric pattern, which has many commonalities to memory, but we can get into that. And the most important property is this: it serves as a set point for the anatomical features, such that when we rewrite that electrical pattern without any genetic change to the cells, they not only build the two-headed worm, but continue to build two-headed worms in the future, despite their perfectly standard genome. So this is just one example.
But just, sorry, Michael, but I don't disagree that if you set up those two things, a purely feed forward versus a feedback set point, homeostasis ever since canon, all the way through to allostasis, no one's arguing about the fact that you can have, thermostats have goal states as well. Yeah. What I'm saying is it's just a massive abyss between that and cognition of the kind that you claim you're going to be able to extrapolate from, which is you explicitly state. In other words, I have no problem showing various flavors of intelligent behavior that come under the general rubric of higher level set points from aggregates that have feedback loops and seem to be able to wander back to the set point from very different parts of the state space when you perturb them. That's all beautiful stuff. And I gave examples: torticollis, spasticity, spinal cord. You have your own favorite ones. There's no doubt that's interesting. But the question is when you say intelligence without a mind and all the examples you give, you have an agenda, which is that you think this is the framework and the way to think that gets us to cognition. You call it basal cognition, meaning, and maybe just two more points I want to make about this. If you read your article very carefully, and I did, you have these surplus words that do your work for you. You say higher cognition. You say more sophisticated. You use these words that are going to do the work of the extra thing that going to college presumably is versus regenerating your liver. You never tell us, well, what is that higher bit, that more sophisticated bit, that more complex bit, that extra bit? I mean, what's the theory of the extra that is going to get us from liver to jet? You never mention it, right? And just one last point about the continuum, which you keep on harping on about. Look, I've used this example on this before. A wing and my arm are made out of muscle and bone and skin. So at some point, an arm becomes a wing. But given that there's no obvious divide, although I would say there is, I could flap forever and I won't rise above the ground for more than 10 seconds, rather like the Wright brothers had to keep their plane up for a certain amount of time. At a certain point, you become functionally useful as a wing because you fly. Before that, even though there's a complete continuum in terms of the substrate, you're not a wing. My arm is not a little bit of a wing. So in other words, we know that you can get functional discontinuities out of, totally, I agree with you, morphological continuities. So just because there's no divide doesn't mean that at some point you start flying and at another point you're not. In other words, you seem to put a lot of truck into this continuity of substrate argument to say, therefore, everything is the same thing. And once I have one bookend and a theory, I'll get all the way to the other bookend. I just don't see the argument being that powerful.
Yeah. Well, I mean, a couple of things. First of all, I should say I in no way claim that I've fleshed out, so when you say like, well, have you gotten to exactly what it is that makes it my decision to go to college? No, of course not. And there are many other, nor do I expect to do this by myself. There are many people working on this anyway. But you see, and even with the flying example, I mean, right? So you've got your gliding squirrels and some things that are, they're not full flying, but you can build a continuum. The real question for all of this is, when somebody like me proposes an invariant, translocation or whatever it may be, you're right to be skeptical, of course, because it's on me to show examples in which using that claimed invariant gets us to a better understanding, more predictive control, a new capability. It has to be helpful. If it's not helpful, then it's garbage and it's of no use. So this is why I say from my first response to the first question is, to me, we throw away any of these analogies that we try on if they do not get us to a better capability that was not available by other worldviews. Out it goes. I'm not interested in creating these things that are not backed up by all of this stuff is meant to be cashed out by actual advances, by novel scientific research programs that did not exist when people were not looking at this with this particular invariant. And so and also I want to kind of push back on this. It's fine to be skeptical about the continuity thesis, but then you've got to offer an alternative. And I have yet to hear. I've had many people say that, well, the human does this, and it's anthropomorphizing to say that other things do it. When you actually ask them to cash out, when does that happen, either on an evolutionary scale or in a developmental scale, there are many things that have no plausible discontinuity. And so it's fine if you don't like the continuity thesis. Then we need an account of, and people will say, well, it's a phase transition, symmetry. More is different, right? That's fine, but then we need specific accounts for those. And I have never heard of a good one for many of the things that we're interested in. People try, of course, but I don't think it's default. And then the continuity thesis is some crazy thing. I think the continuity thesis is taking evolution and developmental biology seriously, and the fact that they're very gradual. And if you want a discontinuity, it's on you to offer a good theory of the discontinuity and where these exclusively human, and of course there may be some, right? Language, propositional language and so on may be one of them. But for many of these things, they go all the way back. If you track it, the thing you are actually interested in, not the shape of the hippocampus, but like the thing that you're actually interested in, goes all the way back in very useful ways.
So I just wanted to say something. It seems like I'm very disagreeing, But Paul Chizak manages to do this kinds of like evolutionary work, but he stays precise, and he kind of compares things in a kind of a scientifically justified way. To me, it seems like what you're saying is that to claim the things that you said as theory, as a scientific theory, is not justified because A, the claims need to be testable themselves. The fact that the claims or the assumptions led to engineering success is not enough for the claims themselves to be valid. They need to be tested against scientific criteria themselves. You can say, I made an assumption, it helped me succeed. For instance, somebody might say, I repeat in my head that I am God, I am God, I am God, and that helps me win a marathon. It doesn't mean that they are God. It just means that, okay, they use this as an approach in order to, and they say, no, I must be God because I said I'm God and then I won the marathon. That success means that this assumption was correct. And I'm saying, no, there are separate criteria to see if the assumption you made were actually valid. And the fact that you think you use this and this is the reason that you won the marathon has the second point of what I was saying. It's the three-part thing. So the first one was, the claim needs to be testable too. We have to test whether I am God is itself valid. Second, could the engineering success be achieved without the hyperbole of the claim? Could a very much more modest assumption or theoretical claim or assumption lead to the same engineering success? I think yes. You could say individually for you needed that, but it seems to me that the hyperbole is not necessary for the engineering success. It would have been enough to say, oh, I use the notion of goals and this lead to that. It doesn't need to say, this is a definition of goals in all organisms across all levels. So I think that's the part where there is the hyperbole. And C, it is imprecise because it doesn't do the required experiments or theoretical moves to support the main claim. So in short, I think I made this assumption and therefore it had engineering success and therefore this is a scientific theory. I don't think that's the way. I mean, based on my years of studying science and reading science and even studying philosophy of science earlier, as well as doing science, I just don't think that I made this assumption and it had success, therefore it must be a scientific truth. That's never kind of the line of reasoning is not for scientific theories. It works for engineering. That's why I give the example of the marathon runner. The fact that the marathon runner wins doesn't mean that they are God. And they say, I won because I assumed I am God. Therefore, that must be true. Or there must be something true. Or I must share some mechanisms with God. But I think the thing that is the point that we are trying to make, and we can move on to other questions. We don't need to stay here forever. And I'm definitely not convinced. The claim we are making, or the thing we're asking for is the assumptions themselves that helped, the scope of them, the hyperbole of them, the justification of them, that seems very questionable. You can give many examples of things that would kind of like say, oh, burden of proof is on you to show me a continuous theory. You can do that, of course. However, the question is still there. The question is something else. The question is, for instance, John asked, how does your definition of goal distinguish between the goal of college going to a kid going to having the goal of going to college versus the liver generating a cell or the frog skin? It seems like it's a little too nebulous. It's a little too, I'm not sure how you're, obviously you can find a way to distinguish it, but that's where the science happens, in my opinion. I guess like we can, please feel free to address this, but I feel like we can move on from this. Everybody agrees, especially I want to hear from Melanie because we haven't heard from her.
I mean, yes, I want to hear from Melanie too. The only thing I'll say to that is look, I may have misunderstood my assignment here. What I tried to do in 20 minutes is talk about a very wide-ranging set of ideas. What I could have done is picked one primary paper and really show you the exact things that you're asking for. I mean, of course, in a primary paper, we have to say, here's my hypothesis, and then you check all the hypotheses, right? And then you show how this works. But overall, I'm not sure what, other than on a broad level, when you have a particular large scale framework, not a very sort of narrow specific hypothesis, but a large framework, other than continuing to generate scientific progress, I don't know what criteria could you possibly have? You know, this God thing that you
So
brought
wait,
up.
wait, yeah,
Red
like
herring.
that.
No, sorry, I'm still
Yeah,
answering.
sorry.
The God thing that you brought up is a red herring, because that one will give you no, you won't get very far with this, right? It's sterile as far as any kind of future progress. And what I'm trying to tell you is that I don't know what else a criterion might be, except for you take a worldview and you see what experiments it predicts. That, you say, could these things have been done with simpler assumptions? Maybe they could have been, but they weren't, right? That's the thing, that nobody had actually done it. And so then you of course dissect how you get from here to there. But what else is a criterion of a useful way of looking at the world? I don't know what else it would be.
So I'm going to repeat what I said earlier in case it wasn't clear. Simpler assumptions didn't happen doesn't make the assumptions correct or true. The fact that these assumptions led to some progress doesn't mean that the assumptions were correct. The assumptions need to be tested scientifically and justified scientifically separately. That's what I'm saying. You haven't shown that. Secondly, we always want people with like bigger ideas. The issue here is not that there were many ideas and very many ideas. The issue here is the imprecision of justifying the assumptions and focusing a lot on the outcome of them and then saying, because we didn't use simpler assumptions, then these are the true ones. I very much think that that's inaccurate, but I'm going to stop here.
I mean, just to be clear, first of all, Michael, the examples you gave and the overview you gave is exactly what we wanted, and I think it does show shockingly fascinating, intelligent, goal-oriented behavior. Where I agree with you, as you said, I'm sure many people never saw that kind of, especially the planaria experiments, right? The double-headed planaria experiments and the memory in them, and they are kind of spooky, to say the least. I mean. And they're amazing. And I can imagine that, I'm not in the field, but you could imagine people being very skeptical and not believing them and not thinking that there was this type of behavior at the level of planaria. So in other words, kudos, and it's amazing. I think the point being made here is the kind of experiments that would have to be done to go from the local assumption, either or strong inference science that was done on planaria or the other examples you gave, to then going to cognition, right? I think that's very hard to do, right? And again, you're not Carl Friston, but you definitely talk about free energy in the article, one of your articles. He explicitly states, and so have others, that this framework will work for homeostasis in a bacterium, and it will explain why somebody decides to go to college. In fact, I think that was the example given in the podcast. Now, would you not agree, or maybe you don't agree, that is an extrapolation too far, because certainly no experiment justifies that extrapolation as of yet? Wouldn't you agree? In other words, it's just how would you ever prove that the goal of going to college and planaria wanting to have two heads or liver generating to a certain size? I think what I was trying to get at is, surely we don't have the experimental evidence to justify that level of extrapolation.
What I would so here's what bothers me when someone says, I'm a human and I have this amazing property of, people talk all the time about decision making. They say, I make decisions. Self-driving cars don't make decisions. Paramecia don't make decisions. I make decisions. I'm a human. When I hear that, what I hear is, I'm magical, and I have these amazing things that only exist in humans. And when I hear that, I think to me the continuity thesis is the baseline. That's the null hypothesis. I want somebody who thinks that the decision to go to college is a fundamentally different thing than the decision of other types of creatures doing other things. I want a fleshed out theory of what that binary distinction is. I have never seen a good one.
But I agree. But let's be very clear, so we don't go around it.
Sorry,
no one's making.
Have you looked for it?
But no one's making that. Yeah, but no one's making.
Yeah, have you studied it? Because I have, and I disagree with you that there is not a clear distinction. It exists for 50 years in computer science, in neuroscience, in philosophy, in psychology. The distinction between goals across different species, even in humans, the different hierarchy of goals. What does it mean to be goal-directed? When can we compare, for instance, even rodent and human goal-directed behavior, and when can't we? So I'm not sure if you're being
when you say I haven't seen any, I wonder whether you've actually looked. I'm not.
Well, I mean,
gonna lie.
what I think he's going to get at, right? I mean, if I understand it, is when you say, just so we don't, it doesn't have to be more contentious, it needs to be. When you say higher level or more sophisticated or basal or more complex, are those words, Michael, sort of continuum words? In other words, there's going to be some qualifier when you say more sophisticated, higher, and you use those qualifiers. Are you saying that whatever those qualifiers end up being scientifically, there'll be continuum qualifiers rather than qualitative ones? Because you too use those qualifiers to say something higher, more sophisticated. So why isn't that special language? I mean, is it really that different saying that we're magical and binary difference, which I don't think either of us claimed, by the way, versus saying we're more sophisticated, which is what you use? Why isn't that another form of imbuing of specialness to just call it more sophisticated?
My claim is simply that so I buy the idea of sort of gray transitions, right? So it's obvious that the humans are a bit different in certain capabilities than other life forms, so that's fine. The question, of course, is what is the control knob that you use to get from here to there, right? And so I think that's a perfectly interesting. So you can imagine, right? So you have a thermostat, and all it does is follow the set point. Then you have a sort of second order thermostat that's actually has a module that can sense when somebody has changed the set point and it tries to fight you on it, right? And then there's sort of one that sets its own set point, and you sort of, and so this is a research program that people certainly do, starting from Wiener, Rosenbluth and Wiener, that try to map out that kind of scale and so I've proposed some steps along that continuum too in that Tame paper. Again, I'm not saying there aren't distinctions. I'm saying that all of these distinctions that there are important and interesting invariants that let you look at unconventional kinds of systems using the same sets of questions you ask about traditional brainy systems, but for example, in different spaces. So for example, one thing I do is I say, look, it is useful to think about behavior in things other than three-dimensional space. Here's an example. Gene regulatory networks. 10 nodes turn each other on and off. That's it. There's no magic. There's nowhere to hide. That's all it is. You look at this thing a priori and you say, well, that's a dynamical system. It's mechanical. There's clearly no intelligence there. I can see all the parts. There's nothing special going on. So we actually took the hypothesis that what if this thing is not as dumb as we think? And we said, why don't we just try some typical behavioral tests that one would use in a brainy system in three-dimensional space? We will try those exact things like Pavlovian conditioning, associative learning, habituation, sensitization. We'll just try those things in its own action space, so transcriptional space. What we found is that typical gene regulatory networks, specifically biological ones, not random ones, so apparently evolution likes this because they're sort of biological ones do it way better. They exhibit lots of the same kinds of behaviors in their own space, and so you can try. They have 6 different kinds of memories, including associative learning in physiological in the transcriptional space. You would never see, and that has massive implications for biomedicine, for example. You would never do that experiment if you thought that cognition and learning only takes place in the three-dimensional world and requires a brain. You just wouldn't ever try training a gene regulatory network. So my point is simply this. Of course, there are transitions, and I'm not saying gene regulatory, they can in a simple way, they can count to 5 and some things like this that you literally can show. We've shown it as published work. You would, of course, there are differences, but that continuity hypothesis leads to you doing the experiment of trying to train the darn thing, which you would never do if you thought that they are a fundamentally different kind of category that had nothing to do with learning. I mean, Absolutely. That's my only claim.
Yeah, right. I absolutely see that there is and forgive me for saying it, sort of remarkable system one-like intelligence almost everywhere you look, and it does dethrone animal intelligence a bit in, as you call, 3D space. It does, right? It does have that destabilizing effect. That's why I think the work is so interesting. I'm not sure, though, that it gets us all the way via this embodiment, via these different dimensional spaces to cognition, which is the, cheeky and fun provocation that runs across the work in other work people who, like Paul. It's that. So in other words, I find myself in two minds between absolutely you've done a democratization of intelligence in other spaces, which is undeniable, and you found it where people probably never thought to look for it. No question. I don't think that success implies some sort of demotion of nervous system intelligence, which I think is the one step,
Oh, I'm with you there. I mean, there's a reason we have brains, of course. I mean, this is I'm not trying to demote brains at all. Brains are great. I'm simply saying that this is not something. I mean, basically I agree with everything you just said. The point is that brains are amazing, and they are using principles that are apparently at play in many other substrates. We should be looking for them. That's the invariant. That's really my claim is just that. There's nothing, I'm not trying to downplay the importance of brains and nervous systems, but that in itself is already, as I'm sure you well know, that's a very controversial claim. There's a lot of people who don't like that way of looking at things. And at that point, all you can do is say, look, it led to new experiments, and we can test specific. Is it really Pavlovian conditioning? Is it really habituation? Well, what are the criteria? Do the criteria say anything about you having to be a neuron? No, it's actually a behavior, a functional criterion. And then you are led to new experiments and new capabilities. That's it.
So Melanie,
I want to hear from Melanie.
Yeah, absolutely.
Yeah, this has been really fascinating. I'll move to a slightly different tack here. Just wanted to probe this notion of all intelligence is collective intelligence, and the difference between sort of individual and collective intelligence, if such a thing exists. So yeah, as an example, at the very beginning, you showed a very cool little video of a single cell. I forgot, a single cell organism that was moving around and reaching out for stuff. At least it looked like it was. And do you think that what you would call the intelligence of that is given up by cells when they become multicellular organisms? Because our neurons don't move around. They don't reach out very far. Typically, they don't. In some sense, they don't have the same kind of autonomy that single cell organism had. So what's the difference between something that feels more like it's part of a bigger individual and something that's its own autonomous entity?
Yeah. Great question. No, I don't think that cells entirely give it up. Although there's an interesting story to be told about what they do give up, which I can say. Keep in mind the thing I think again that gets us here is that we are so conditioned to look for autonomy in three-dimensional space. So true, neurons don't move around the way that this thing does. But if you look at behavior in transcriptional space, behavior in physiological space, and behavior in anatomical space, lots of our body cells, including neurons, still exhibit some amazing various capabilities. So, for example, there are people studying diabetes as a learning disorder in the pancreas. There are immune system cells that learn not by this kind of selectionist. I mean, of course, that also happens, but not because of the selection thing, but they actually learn from experience. There are cancer cells that habituate to chemotherapeutic agents. The story that I told about gene regulatory networks, that goes on in every cell in your body, right? So they do get bound to a common purpose in a particular way, especially in with respect to morphogenesis, but they are still doing many things to navigate their problems, various other problem spaces, physiological, metabolic, and so on, with various competencies. Some of them as simple as habituation, which is a huge problem. Drug habituation, right, for pharmaceuticals is huge because physiological pathways habituate just like brains do, and so on.
Okay. Let me ask. I understand. Let me ask. Just why if you cut my arm, it won't grow back, right? And if you liquefy my brain, it won't.
Sorry, Melanie, your voice is
your voice is very robotic, and that's why the feedback of Michael's voice was getting robotic. Do you have another microphone? Something happened. Maybe the wire. Is the wire loose? Because it was okay before. Now you're muted. If you unmute now. Now you're muted. You need to unmute. If the mic is not attached, it might not unmute itself. I'm so sorry. Where are you gonna? Oh, I unmuted you multiple times. It's oh, I see. If you disconnect a mic, it sometimes doesn't let unmute. It's happened to me before. You would need to go out and come back in, I think. I think that would be better than typing, because this is a very interesting discussion. Today, microphones are on strike, it seems. This is the second time we're having microphone issues. Also, very cute dog over there. Okay, let's see if she's back. Okay, I'm just gonna invite her again. She's connecting now. And Michael, I saw that you need to leave soon. We usually go longer. Let's see if we can wrap this up soon. Melanie, go ahead.
Yeah. Sorry about that. I think, yeah, I was asking, like, why doesn't my body have the same kind of flexibility, plasticity as planarium? And you're muted now, Mike.
Okay, can you hear me now?
Yeah.
That's weird. Yeah. So okay. So that of course is a multi-billion-dollar question. Many people are trying to investigate this very issue. Nobody knows. I have a hypothesis that I can sort of trot out, but nobody knows for sure. So and we can think about it at two levels. One reason why, so if you think about it, with only one exception, all the highly regenerative animals are aquatic. And the thing is that it's not that they're simpler animals. So there are many species of salamanders that don't regenerate, and planaria that don't regenerate, and ones that do. It's not that, but it's not the sort of supposed simplicity of them. There are kind of ecological trade-offs. So imagine our ancestor as a mammal, right? So you're running around the forest, you're like a mouse-like creature, somebody bites your leg off. Here's the problem compared to a planarian or a salamander. First of all, you're not buoyant, and so anything that tries to regenerate on that limb is going to be ground into the forest floor. It's not going to regenerate. It's going to get infected. Also, because it's not an aqueous environment, you can't run any of the bioelectric injury currents out of the wound that you normally need in order to regenerate. And so one thing that we might imagine is that what evolution decided, the solution evolution found, is to shift you to scarring. And that, in fact, is what mammals do, is they seal the wound, they scar, massive inflammation. Let's just not get infected and hope to live another day, right? It's, you're never going to get a chance to regenerate anyway. So that's a typical story. We don't know that's what happened. Now, one of the things that we work on, of course, as do other people, is to try to induce regenerative response. And so we have had some success on it, as you know, and the field is moving forward. I don't think I don't think it's fundamental. I think we are going to be regenerating. I mean, we are certainly working on limb regeneration in mammals, and I think someday it'll get cracked and we'll be able to do it. But the more fundamental, so I have a more fundamental kind of idea about this. One of the things about planaria specifically is that when they reproduce, they tear themselves in half. Each half regenerates, and now you got two worms. That's how they reproduce. That is completely different from us in the following way. We have Weissman's barrier. So when we have a mutation in our body somewhere, our children don't inherit that mutation. In planaria, that's not true. So because every worm is repopulated from a piece of the adult, anything that doesn't kill the stem cell propagates into the next generation. So if you actually look at their genome, they're an incredible mess, and they're mixoploid. Every cell might have a different number of chromosomes. They look like a tumor. I mean, it's just an incredible mess. And yet, these animals have perfect regenerative capacity, extremely robust, very cancer resistant, no aging. They're immortal. Why would that be, right? With the worst, I mean, isn't that kind of a weird thing that the animal with kind of the messiest genome has the best anatomical properties? Like, that's really weird. So again, nobody knows, but I have an idea about this. So let's imagine for a moment that you have a creature where so typically people think there's a genotype and then there's a phenotype, and the phenotype is what gets selected. But there's this layer in between, and this is the stuff we've been working on. This layer does interesting things, like, for example, if you're a tadpole and your eye or your mouth starts off in the wrong position, it will either move to the right position, or if we put an eye on the tail, those animals can actually see, right? With no period of adaptation, they can just see. And they do great in visual learning assays and all that. So there's this period of developmental rearrangements that can overcome quite a lot of defects, bisection in half in the case of like early embryos and so on. So what that means is, so you've got some amount of competency to fix various kinds of errors, right? When you come up for selection selection doesn't know whether you look amazing because your genome was amazing, or whether your genome was actually so-so, but the competency was really great and it fixed up a bunch of stuff, right? Was your eye off to the side? Who knows? So what that means is that a small amount of this kind of developmental competency hides quality of the genome from selection. And so at that point, evolution has real trouble picking out the best genomes. But what it can do is pick out the most competent, select for competency. Competency ratchets up. Well, that makes it even harder to pick genomes. And so there's a positive feedback loop where different lineages sort of drop off at different places on this feedback loop. But what I think what happened in Planaria is that it went just as far as it could go, meaning all the effort went into developing an algorithm that builds a perfect worm, no matter what the genetics looks like, plus or minus. We've done other experiments that are unpublished that are just nuts, culturing these things in mutagen for a year. I mean, another funny thing about Planaria, every other model system, so mice, Drosophila, humans, you can get mutant lines. You can get, fruit flies with weird eyes and wings, and there is no such thing as a mutant line of Planaria, other than our two-headed worms, and those are not mutants. They just don't, the genetics of it just don't make that much of an impact, because I think evolution has basically done most of the work on this kind of algorithm that's incredibly robust to hardware defects. So I think that's what's happening there now. Salamanders sort of stopped earlier, and mammals kind of didn't even go that far. That's one way to start to think about this, right? Is like these error correcting codes that basically allow you to tolerate all kinds of junk at the hardware level. So we'll see. Yeah.
Okay, I should let John Cubey ask his question since we don't have much more time.
Hi, really enjoyed the talk.
Hey, John.
Getting back to difficult issues, I asked a question initially about agency. Is agency meaning the individual itself sees a goal, or that we as an observer can see that the Planaria is going towards something? I think that's a very, very different thing. You, in your final slide, seem to deal with that a little bit, but it skipped by very, very quickly. I think your final slide seemed to me contradict some of the things you were saying, but I'm not really sure.
Yeah. I think that so I think that's a very important question. So all of it is observer dependent, meaning relative to some observer, except that certain systems are also such observers that can make those hypotheses about themselves. So in the vast majority of the systems that we look at, the human scientist, of course, is the major observer, but in certain senses about certain things, the system itself is also such an observer. And in humans, we have a very complex metacognitive capacities to tell these stories about ourselves and our own goals and things like that. But none of the kind of homeostatic and allostatic models that we deal with require that kind of second order I know that my goal is whatever. I mean, I'm not saying planaria know that at that level. Of course they don't. We are the observer that formulates a model of an allostatic model of what the planaria is doing, and we test that model in various ways, and it helps us or it doesn't.
Isn't that a discontinuity to being an internal observer versus?
So I'll tell you. Here's a story that this is due to Chris Fields first put me onto this. I think it's a very interesting story. So you have a bacterium, and the bacterium is wants to increase, let's say, sugar concentration in its environment, and it's measuring the sugar concentration, and it's going up or down the gradient, and so on. So you can do that, but what if the sugar molecules are poisonous? So one thing that some bacteria do is they don't measure the sugar concentration, they measure the output of their own metabolism. So at that point, now that's interesting, because what you've done is you've coarse-grained a bit, so you don't know exactly what all happened as far as was it sugar, was it not? But what you do know is how are we doing metabolically? That's what you know. And so I think that it is a discontinuity in the sense that you have to use a slightly different model for that bacterium, because you have this like meta, sort of this loop that monitors your own state as opposed to being focused out into the outside world and purely sensing the outside world. So that's a different model you have to use. But I do think that is the sort of thing that kicks this all off and running to the idea that, yeah, we have sensors that look out, and we should also have sensors that look in. And when we make models of the outside world, we can also make models of ourselves within that world. So there's the continuity.
There's a second introspective discontinuity, I think, that I can imagine the future. And I can say, if I do this, this will happen. If I have a sense of time in the future, and I don't see how that extrapolates all the way down.
Well, so I'll say two things to that. I'm certainly not claiming that our human ability to time travel far into mentally time travel into the future is exactly the same in other simpler organisms. Okay, so I'm not making that claim, but here's what I'll say about continuity and discontinuity. So that pattern that I showed you let's assume, here's what I like to do for all these kinds of complex human level things. I would like to ask, what is the simplest possible version of it? Like, what's really the core of it, right? So what could be the simplest possible version of whatever? So let's ask, what is the simplest possible version of a counterfactual cognitive state? Something that you can hold that isn't true right now. So I'll tell you two kind of cool stories about that. In planaria, what we can have is we can have a bioelectrical state that indicates what you are going to do if you get injured in the future. Not what you're doing now. Nothing about it doesn't reflect anything that's true right now. It's a bioelectrical state that says 2 heads. You don't have 2 heads. You have 1 head. So it isn't true right now. But it's a sort of aspirational future state that tells me as a scientist, I know what you are going to do when you get cut. It's the exact same thing when Tanagawa and those folks look at a mouse's brain with optogenetics. They know, at least in some cases, they try to figure out what is that mouse going to do by reading states that are not true now. There are things that guide future behavior. So this is a very simple, very basal version of how does a collection of cells hold on to a piece of information that is not true right now. That if I had to come up with some sort of like, what's the evolutionary precursor of our amazing human time travel, might it not be something like this? And there's an even simpler version of that, which is Audrey Dussautour's work on Physarum, on slime molds. And what she has done is she has shown, so you got a slime mold, and you put a piece of you put a salt stripe next to it, doesn't like it, doesn't cross the stripe, then it gets hungry, crosses the stripe, and it finds food. So you do this 5 or 6 times, and eventually it will cross right away. It just learned that it's worth it to tolerate the salt to get the stuff. So in a recent paper, she asked, what is that? What is the memory trace in this? This thing is 1 cell, right? It's a unicellular organism. What is the memory trace that allows it to remember that crossing salt is okay? What they find is that it literally holds some salt. It basically imports the salt, right? So even better than having some kind of a symbol of something that isn't true now, the most simple version of that is having literally a piece of it. Like, it literally has a piece of that salt. That's my token for recognizing what this is, right? So you can build up. I think you could sort of build this up. But to me, the question isn't, do Planaria and Physarum imagine the future the way we do? Well, of course they don't. But what could be a simple, a scientifically useful, simplest version of what's interesting about that time travel? And to me, it's the ability to hold state about things that are not true currently. That's how I would boil it down to what's essential.
I basically sort of agree with you. Even the wing analogy, nothing springs from nothing. Things don't. You don't have suddenly have a human-like representation of the future evolved out of nothing. It had to come from something, and finding the roots is a terrific task.
Yeah.
Do you want to go next? Zhuqian, am I pronouncing correctly?
Yes. I would like to. So I have two questions. The first is that when you talk about collective intelligence, like on a neuron, like, so when you talk about collectively, let's take a brain or like a morphogenesis. So you say like a neuron can have a goal, like the bacteria can have a goal, and then when they are taken together to form like some kind of collective forms, and they also have a goal in some ways, like the neuron, like for instance, saying like the neuron have its own goal and the brain has its own goal. So how do you see this discrepancy, like from like, it's like basically you're saying that we can understand each level of the hierarchy having their goal and their own intelligence. So that how does that go collectively to form it's not intuitive, like when the neuron has its own goal and the brain has its own goal and they come together and coherently. That's my first question. And the second is like engineering wise, that you can manipulate like some of the features and then they generate really interesting behaviors like xenobots. But I'm asking like what, like on an ethical level, like would you not have the problem, just like when you deal with deep learning, the unintelligibility of what happens, like when you have these kind of manipulations and you have those kind of like behaviors, although they might be useful, but you do not understand on a level that what actually happened in a genetic space or a transcriptional space, like if you don't go even further down.
Okay, a couple of things. With respect to so what I think happens when things scale up is two things. First of all, the size of the scope of the goals enlarges, and also the space within which useful goals can be found changes. So single cells have metabolic goals and transcriptional goals. Collectives of cells can have anatomical goals, and brains can have behavioral goals and maybe linguistic goals and whatnot. For sure, we do not have the ability to predict those goals. This is the future research program, is to say, if we understand the goals of the parts, can we predict and control the goals of the collective? This is exactly what we are studying. We only understand this in very few cases, if at all. That is the big mystery facing us, is where can we learn to predict and modify goals of these collective systems? So no, I don't have an answer for that. That's what we work on. With respect to understandability, so I'll give you a simple example. So skin cells from frogs becomes the xenobot. We did an experiment to exactly what you just said, to ask to drill down and see how is it working in these various spaces. We did a transcriptome analysis, so we looked at all of the genes that are expressed by these xenobots versus all of the genes that are expressed by frog skin in the native context. We found out that the xenobots express, I don't remember the exact number, but it was well over 1,000 new genes that standard embryonic skin never expresses. So of course, now one of the things that we're doing is we're analyzing, okay, so these cells found themselves in a new environment, a new context. They're swimming around like a ciliated larva of some other organism. So you could do a simple bioinformatics experiment. You could say, among those new genes that it's turned on, right? Does that transcriptome look like other creatures, like, for example, a marine larvae of various sorts that live the way that xenobots live? Did they turn on those kinds of genes? Because that's now the type of animal that they are, even though the baseline genome is exactly the same. So that's a typical experiment. I mean, that's not the only thing, of course, but a million things to do. But that's a typical thing that you might do. You might look at how how it solves that problem. I'll give you another example in planaria. What you can do is you take planaria and you throw them into a solution of barium. Barium is kind of a non-specific potassium channel blocker. It blocks their ability to pass potassium. Their heads literally explode because all the neurons are hyper, overexcited, whatever. Their heads explode. If you leave them in the barium, within a couple of weeks, they regrow a new head, and the new head is completely barium adapted. No problem. They live in the barium fine. So we asked this very simple question: what's different? What's different about a standard planaria head? What's different about the barium adapted head? There are many things that could be different, but one thing we looked at was transcriptome. So we just looked at what are the genes that are different. There's only a handful of genes out of the whole genome that this system was able to up and down regulate to allow them to do their physiological business despite being in a high dose of barium. And so now we have to reckon with the fact that planaria never see barium in the wild. There was never selection specifically for barium adaptation. Now, there might have been selection for ways to deal with hyperexcitability and epilepsy and who knows what else. But one thing that you then want to understand is, what is that walk in transcriptional space? You start as a planarian with normal gene expression. You need to get up here. It's a very high dimensional space. Many of the things you do will actually kill you. So you can't just do gradient descent. You don't have time to sort of randomly poke around. How do you get from where you are now to this kind of just a few genes being the right genes being altered? And what is that step? So now you can do single cell RNA-seq. You can compare across animals, across cells within the animal. That's the research program. But it's driven by this desire to understand navigation in transcriptional space. And again, you wouldn't do this experiment if you didn't think of these things as navigating space to solve problems. You just like that. You wouldn't, I have colleagues in autonomous vehicles who are giving us some amazing, ways to think about how you know where you're going and what the error correction modes are and so on. Like, these are fruitful ways to do these experiments, but of course you have to do the experiments, which we're doing.
It seems that your understandability lies in a genetic level. Is that you seek on a genes level or cryptome levels? You can explain in terms of that. Yeah. So I'm wondering that. But. So that's at the end of the day on a reductionist approach to the genes. But when we talk about brains, the genes does not tell you about brain intelligence. So right. When you talk about all these kind of interesting behaviors on this planian, so you're talking about the intelligence is in the gene, or are you talking about but how does that differ? If at the end of the day you so where it goes down, but in the brain, genes do not tell you anything about the intelligence of the brain or human intelligence. It's
well, I don't know if they don't tell you anything, but I mean 100, but I'm 100% in agreement with you that the traditional models that are taken to be explanations in the molecular biology literature in terms of genes, I completely agree with you that they're not the interesting thing here. So our models tend to be computational models of information processing. The transcriptional readouts are the behavior. That is the behavior. That's in this case. We look at many other things, physiology, anatomy, anatomical space, whatever. But in this case, the changes of gene expression, that's the readout. That's the behavior. What we want to understand is, how do collections of cells turn on, take steps in that space. In this case, it's a transcriptional space. In other cases, it's an anatomical space. I can give you some more kind of amazing examples of them doing it. And strictly speaking, yeah, we've had many studies where we show that the genetic information is completely overridden by the physiological information that the system processes over time. The genes are not the answer. What the genes do is provide some amazing sort of low-level hardware that's very good at solving certain kinds of problems. So that's great. So you need to understand the hardware, and then you need to understand the algorithms that it's using to solve various kinds of problems. And by the way, what problems can it solve? We still, I think we haven't even scratched the surface of what some of these things can do, because standard development and standard physiology is puts you into a very false sense of kind of this is how, of course it's this way. How else could it be? But when you put things in very unfamiliar environments, you really get to see some of the limits of the plasticity, and then you find new competencies that they have.
So
Michael, can I ask you a question? Oh, sorry, Hokin. I just wanted to ask, just following up on that. I mean, because you said, Hokin, I think it's very important that you hear that what Michael is saying is precisely not at the level of the genes, right? In other words, that's exactly what isn't happening, right? In other words, it defies that hardwired genetic belief. You see this incredible flexibility in the setting of an unusual perturbation. And I was just thinking, Michael, would it be correct to say, because we're finishing up here, that do you think is there a principle? I'm not sure if you've explicitly stated it in the papers you sent, but I'm beginning to see it, that if you have a living system that has to live in a world that has some noise in it, that this kind of nested hierarchical structure where you can have goal setting and movement, problem solving through the state space of this higher level is a general life principle of intelligence. In other words, that you basically, that there are these different state spaces, whether it's transcriptional or whether it's, spatial, and that the solution that keeps being recycled, and I think you're talking about this incredible recycling notion of a principle, is a representational level above the level of genes or any other building block that can give itself a self-regulated feedback goal cycle that can actually move through that space in an adaptive way to the external world. In other words, it's almost like a universal principle. It's that one, right? That it's somehow this ability to set up a state space with a learning rule or a feedback rule that allows it to move through that state space adaptively depending on the world changing. And it seems to be that keeps coming up in all your examples. Is that essentially what intelligence is for you?
Yeah, I mean, I take that question, you can tell me if I've understood, to be really asking about what the Venn diagram is between the set of intelligent things and the set of living things, right? Is that really what we're talking about?
And it seems to me that they're almost the same thing for you, because you've given so many examples in every living example you've given.
Yeah.
And it almost seems to be that problem solving through the selected space to fit the environment's perturbations.
Yeah.
Almost seem, and I think you have somewhere in one of your papers the example that, a stream finding its way to somewhere, it may look like it's negotiating the rocks and the pebbles, but it isn't, right? You call that flat, something like that. So is that basically, they seem to be coterminous for you, the definition of life and that problem-solving space that every living system seems to be able to set up.
So here, okay. So I'll address the kind of Venn diagram perspective here, and so I'll, at the risk of saying things that I think you'll find even less palatable than some of the other stuff I said. I think it's worse than that. I think it's not just that they are sort of overlapping. I think that there are a wide range of potential and some actual things that we would not call alive that I do call intelligent and cognitive. And I think at this point, I don't believe that there are any living things that are not somewhere on the cognitive continuum, although possibly at the beginning of the origin of life and maybe in synthetic biology, maybe we can make some things that I would say are not on the, I actually don't know. I don't have a firm opinion on that. But now, looking, with life as it is, I think every living thing has some degree of competent problem solving. I think everything that didn't, it was, basically killed off long ago, if it ever existed. But I do think that, so just to give you a simple example, a chemical pathway, as we've seen, is capable of 6 different kinds of learning, and also this is unpublished, but we have some very recent work where they can sort of go around barriers in that space and do some interesting things. I wouldn't call that alive. Now, I should also say that I do not have a definition of what it means to be alive. I spend zero time thinking about the definition of life, unlike some of my colleagues like Sarah Walker and Lee Cronin and people like that. So I don't know. Maybe there's a useful definition of life that does overlap exactly with what I'm talking about. I'm much more interested in diverse intelligence, and my current opinion is that there are things that are intelligent that we would not call alive. But I also don't hang my hat on any particular definition.
And just to finish it,
Sorry, 1 second, a time check. Just a time check. Michael, you wanted to go a while ago. Just wanted to check with you how much time you have.
Yeah, that ship had sailed out. So let's do another 5 minutes, and then I'll.
And would you say then that,
Okay,
the current sort of AI gold medal winners, whether it's the transformers, that the generative models, or what deep neural nets have been doing, do they have this kind of intelligence that you are claiming a kind of universal principle for across the spectrum in your papers? Are they doing intelligence in this way or not, do you think?
Yeah, so prefacing this by saying that I'm hardly an expert on how all the details of those architectures, so I don't know everything about how they're made. I wouldn't rule out that they're doing something that fits the definition, but I believe they're missing a number. And I tried, we had no time to talk about it, but on one of the last slides, I tried to list some of the things that they don't do, which I think is very important in the generation of a true agent. And I'm currently working on a paper that outlines what those things are, right? So what are the requirements of an agent coming into the world in a more biological sense? So I think there are many things they are not doing. So I think they're quite distinct from most of the kinds of things we're used to dealing with. But that's not to say that they might not exhibit in their own space a form of intelligence that meets my criteria. They may well have, but there are major differences in how they come to be, with respect to not having energy limitations that they need to deal with a fixed boundary between them and the outside world, and various other things that this struggle, right? Somebody said, I don't remember who it was, maybe Eric Hole or somebody said that the shared struggle is really important, and all living agents share this struggle from moment one of where are my borders, what effectors do I have, what sensors do I have, what is a proper model of that's going to maximize my adaptive utility. As far as I know, these things don't have that. They don't have to try to define themselves and make self models. I could be wrong, but and that's not to say we couldn't build something like that, right? So I think if we understood the correct things, we could totally build something like. I don't think evolution has a monopoly on producing intelligent agents at all, but we will have to do that. I don't think we've done it yet.
So if I were to look at a graph of superordinate, just so I understand, we have agents, we have intelligence, and we have life, right? And I think actually Kevin Mitchell has a book coming out called Agency, and you have it. So the superordinate term for you is there's a particular kind of intelligence of the kind that you've been outlining beautifully that is really agential. In other words, they have barriers between themselves and the world. They have to continue to exist. All agents will have an intelligence with this general basal cognition that will then extrapolate all the way to us, you hope, right? But then there may be intelligences that are not along that evolutionary pathway, that are agential and have basal cognition. They may be other intelligence that are non-agential. That's possible, right? But you're talking about agential intelligence.
Yeah, I think that's fairly close to what I think. I put not very much emphasis on what you're made of and how you got here. So I think, natural evolution is fine. Design is, human engineered design is fine. Any combination thereof using, evolutionary algorithms and whatnot. I don't think any of that really matters. I think there's a continuum of the kind that Wiener et al. First put out that enables you to use the same techniques no matter what you're made of or what space you're operating in. And these are techniques, they're functional. You cannot make these decisions by looking at things. So no amount of observation, purely observation of data tells you anything about the intelligence of something. Many things look like they're intelligent and they may not be. But there's a set of techniques, perturbation techniques, where you get to ask what are the competencies in some defined space, and each of us is free to propose some other space and see how we do. What are the competencies? And I think that applies broadly, no matter what you're made of, no matter what your sort of material implementation is, or how you came to be. And that's the critical part, is how you do under that suite of tests. You know,
Mhm.
and we've literally, I mean, we literally published and it's nothing new. People have been doing this kind of thing, except we're now proposing you can do this to other systems that you never thought of. These tests exist in behavioral science. They've existed for decades, but you can apply it to all sorts of crazy stuff that nobody had ever tried before.
So in the little time that's left, I just wanted to make sure we read a couple of audience questions so that we make sure we don't sort of skip them, if that's okay with you. So quick answers would be great, or however you feel like it. We want to make sure that you go as in time, and just let us know 1 minute before your, chariot turns into pumpkin. So one question is, to my mind, near criticality of brains seems part of what makes them so magic. Do you find using tools for neuroscience that in these other systems?
We're certainly looking for it, including other signatures such as integrated information, various other metrics. Stay tuned. It's not published, so I'm not really. I don't want to really say much until it's done and peer reviewed.
Another question is, sorry, this one. Guru was interested in the example of caterpillar's metamorphosis into butterfly, and they ask if there are studies to further understand what is conserved in the memory across caterpillar and the butterfly and in other features, and where those memories reside.
Yeah, it's quite unclear. The whole, there's a huge field, of course, of where memory resides in general, and there's all kinds of controversies about whether it's synaptic structure, or it's RNA, as Glanzman thinks, or it's cytoskeleton, or what is it? No, it's not known. What is known from the planaria, about the only thing we know from the planaria example, is that behavioral memories can be stored outside the brain and somehow imprinted onto a brand new brain to drive behavior that's consistent with previous memory. That's about all we know. The mechanisms are not clear yet. Sorry, I think you're muted.
I just wanted to say there was also the example of a worm eating another worm and having the memories of the worm it digested. So that was also
we did not do that work. So McConnell McConnell discovered this stuff in the 60s. Now he was right about everything else, so I don't see any reason to doubt him. But we did not reproduce that. The other thing we did ourselves using an automated paradigm in 2013. So he was completely right about that.
Did you try and fail the replication, or no? It's just
the feeding thing.
Yeah.
We never tried it. We never tried.
Okay, got it. Just wanted to check. Another question is, if we were to make xenonauts from other cell types or species genomes, would they develop the same cellular behavior?
Stay tuned. In about a month, you will see the answer to that. I can't go into the details, but the bottom line is it isn't the genome. That's kind of what I'll say at this point.
And I had invited them to ask this question online. Thomas Ryan asked, and they say that they accidentally clicked no. Where do you think the organismal information lies? At the structural, developmental level, or do you think that it would be a way of quantifying or isolating it?
I'm not sure I understand. What is organismal information? Do we mean anatomical shape?
I'm not sure, because they, unfortunately, I don't know if they're here. There's going to be a delay, so I don't know. But if they just say organismal information lies, I guess I'm not sure what they mean, to be honest.
I'm sorry, I'm not sure.
Another okay, so let's see. Another question was by Jamie, why are brains so big? Could a brain be made with fewer neurons, less energetically costly? I guess in response to your idea that these are all the same functions.
So I don't know. I mean, it carries so much, metabolic cost and other costs, at least in humans during childbirth and all that, that there must be some reason why it's huge. But I will point out to one. I'll just point out to one kind of weird thing. In the 1980s, this guy named Lorber published a paper called "Is Your Brain Really Necessary?" And what he was looking at is very rare examples of patients with severe hydrocephaly, where the brain was just a very. I think the patient had the third of a cortex of a chimpanzee, but above normal human intelligence, and so. There must be, so I don't know what to make of that. There must be some reason why we have these giant brains, but there are individual cases that appear to do human level intelligence, in a normal way. So I don't know, I don't know.
Well,
we don't think there's human level intelligence in those smaller organisms. At least there are some intelligence that doesn't translate. Another thing is someone who, Thomas Ryan is clarifying. They say they mean information at the phenotype. I'm not sure still what that means.
I don't know what that means. There are many different kinds of phenotypes, right? Anatomical, physiological. I don't know.
Another person says, where does intelligence reside in cells?
Yeah. So there's a few places. So for sure, the pathways and the gene regulatory networks can do some things like 6 different kinds of learning. They can count to small numbers, things like that. No doubt there are other mechanisms. So people, Tazinski and other people like that have looked at computations done via the cytoskeleton. So there's probably that going on. Who knows what else, right? I don't know, but cytoskeleton and molecular pathways are 2 for sure media that could do it.
And Sukyan didn't ask this question, but it got very highly voted, so I'm just going to put it here. Is it whether your view is close to panpsychism?
Yes, but I, in the last 4 minutes, I'm not going to be able to do a defense of panpsychism, obviously, but I have some sympathies for it in an important way, for sure.
That's consistent. Thank you for that. I think Thomas Ryan, as the last question, clarified their question. They say, do we think that information may be embedded in the shape or topology or the organism? And this allows for regeneration to maintain structure, ?
I'm not exactly sure what that means, but we know that many organisms can regenerate structures that are completely missing. So it's not as if, like, I've cut off one finger and thus I'm going to just copy it into from the other fingers, right? We can, in salamanders, we can get rid of the whole limb and just everything.
I think that their issue is between genetic versus something else. But to be honest, I don't understand completely any of the sentences. So I don't think we should kind of try it again. Thank you so much. I just wanted to say, we discussed like a little bit, but it's the nature of the salon that we like discussion gets like more into the nitty gritty, but I really appreciate the big picture idea. And I'm very sympathetic to it. I personally would be very interested in mycelial intelligence, which is not even close to mammals or like worms. It's not even an animal species. It's not a plant species either, somewhere in between. It's ancient. Very much. And I do wonder whether one of my dreams in writing science fiction about it is that at some point we can build AI not with silicon but in mycelial networks that we can upload information to. So I'm very sympathetic in the kind of the bigger vision, even though I disagree with the extent of the claims, and I think some of them need a lot more backing. Or you can say, I'm not going to back this up. I'm going to make this assumption for engineering purposes. I would very much be kind of like sympathetic to that as well. That's very fair. I think the point where we disagreed is the validity of those as claims of themselves, as opposed to, I guess, what Wittgenstein called a ladder that you climb and then throw away when you've reached the top, so to speak.
Yeah, I'll just say also to thank you that what I do think, just the big picture, and tell me if I'm getting this wrong, is it's very true that when it comes to thinking about intelligent behavior, there absolutely has been a bias towards nervous systems and certainly towards higher order nervous systems. I think that's true. And I think what you're getting at, which I think could very well turn out to be true, that if you have a far more ecumenical, far broader notion of intelligence as problem solving in representational spaces, and you do look at intelligence in all the beautiful ways you've shown it, morphogenetically, transcriptionally, wouldn't it be delicious if that was the better vantage point to go back and see what was the extra that nervous systems did? In other words, that it was a better origin to get there, and where we think there are differences is from your platform, from this platform, rather than going directly to the nervous systems and thinking that they're going to reveal their secrets outside the context of this broader definition of intelligence. And I think that seems to me what you're really getting at. And I think that's may very well be true.
Yeah, we're going to have Melanie. Sorry, I just wanted to say Melanie might have the last question after what you're saying. She's going to say something and we're just going to close the session.
Oh, okay. Well, this was to clarify the question from Tomas about where the information is, for this regeneration phenomenon. Is it? I think he asks, is it? If we're talking about the structure of the organism and it's not in the genome, where is it?
So I don't. Do you want an answer or?
Yeah.
Because okay. So I showed you one example, right? So, and of course, this is just the tip of the iceberg. If you want to know where the information is for the number of heads that you should have as a planarian, it is not in the genome because so I've shown you this. It is literally in the distribution of bioelectrical potentials that is rewritable. Now that we figured out how to do it, it's rewritable like any good memory. It's labile. That is where the memory is. What the genetics does is build an electric circuit that by default reliably lands in a pattern that says build one head. We can shift it to other, and the thing I didn't show you is we can also shift it. So that's the number of heads. It also sets the shape of the heads. And in fact, what we can do is shift it to build heads of other species. We're using the exact same genome. So we can make worms with heads of 3 other species using the exact same thing by pushing it to other memories that it could have that it normally never has. It's very reliable, but we've learned to push it that way. And of course, this other issue that we could also push it further to make shapes of things that don't look like flatworms at all out of flatworm cells. They look like weird hedgehog shapes and all kinds of other stuff. So that's where it is.
So is there something, I'm sorry, is there something like that also going on in the caterpillar to butterfly memory?
That I don't know. That's a perfectly reasonable hypothesis. All I know is this, that I know it works in one direction where we can take, for example, memory blockers and use them to wipe that non-neural bioelectrical pattern and make these cells forget how to build a planarian. They become featureless circles that have no idea, they just don't make metamorphosis at all. I know it works in that direction. I don't know if it will work in the other direction, but I'm hopeful that it probably will.
That was actually very cool. Thank you so much. One more thing. I'm so sorry. TXC is asking very interesting thing about your approach. And they ask how you embrace students who have other conceptual, technological, analytical ideas. And how do you feed back and forward and unlearn, learn together, and other lab members? I guess they're asking this because of the creative nature and kind of unconventional nature of your research. So maybe it's not bad to close with this to appreciate your unconventional approach.
Yeah, so my group has a variety of people with different temperature settings as far as how unconventional they want to go, and I support whatever people want to do. So if they want to work on something that's a conventional mechanism, if it helps us to understand what we need to understand, great. And then I have other people that have ideas beyond what I sort of can think about. And as long as we can formulate a testable hypothesis that gets us to a new research program, then we do it together, and then it's fine.
Very much appreciated. Thank you so much. This has been a fantastic session. Sorry to hold you for 20 minutes longer than you wanted to. I really apologize.
Thank you all so much. Great discussion.
Thank you.
Have a great weekend.
Thank you.
Bye.
Bye.